4 bottles, 4 very different whiskeys. Today, we're putting every one of them to the test. First, we'll see what they leave behind under the microscope, and then we'll introduce each one to a living nematode and see how they react. So I've got 4 of the most popular whiskeys sitting here right on the workbench. Fireball, Maker's Mark, Bulleit, and Jack Daniels. I'm labeling them sample A, B, C, and D, because I don't want the names influencing what we think we're seeing. By the end of this video, we'll reveal which sample was which. So before we take a look, I'm adding a tiny drop of methylene blue to each sample. Since whiskey is almost completely transparent, the dye helps increase the contrast under the microscope, giving us a better chance of seeing any tiny particles or residue that might be there. Okay, so let's start at 40 times magnification and see what sample A has to show us. Okay guys, so when we're looking at whiskey under a microscope, you're not looking at the alcohol itself. Alcohol is completely transparent, so you can't actually see it. What we're really looking for is any microscopic material that's suspended in the liquid. That's why I added the methylene blue. It gives us enough contrast to actually see what's there. So as we zoom in, notice how that blue material isn't spread out evenly. It's collecting into little patches across the slide. Those patches are made up of microscopic material suspended in the whiskey. Exactly what every particle is, we can't tell just by looking right now through this microscope, but it gives us a really good baseline to compare against the other samples. So at 400 times, take a look at this. You can really start seeing how fine this material is. And what you're looking at are the tiny microscopic particles suspended in the whiskey. They can come from things like barrel aging, natural organic compounds, or other ingredients used during the production. That doesn't automatically mean it's good or bad. It just gives us something to compare to. If another whiskey has a lot more suspended material than this one, we'll probably notice it pretty quickly. All right, so now we're at 1,000 times, and this is about as close as we can get. You can see that these tiny particles aren't all the same size, and they're not spread out evenly either. That's exactly why I like comparing samples this way. The microscope isn't telling us which whiskey tastes better. It's just simply showing us differences that we'd never be able to see with the naked eye. So now let's see if the next sample looks the same or something completely different. Okay guys, here's sample B at 40 times magnification, and right away you can see these larger clear areas with tiny little circles inside them. Those aren't alcohol molecules. What you're seeing is how this sample spread across the slide after I added the methylene blue. So every whiskey seems to behave a little differently, and that's exactly what we're comparing. So as we zoom into 100 times, you can start seeing more of these tiny round pockets. They're not all the same size, and they're definitely not spread out evenly, but that's one of the things I'm paying attention to. Does every whiskey organize itself the same way on the slide? Or does one stand out from the rest? So take a look at this of 400 times, and the pattern almost looks like a honeycomb. Those little circles are tiny liquid pockets that formed as the sample settled on the slide. They don't automatically mean this whiskey is better or worse than the last one, but they do give us another way to compare how each sample behaves under the microscope. So before we move on to sample C, what's the difference between sample A and sample B? Did you guys notice anything drastic, or is it the same? Okay, here's sample C. Okay, here we are, sample C at 40 times, and right away you can see these large clear areas where the liquid has spread across the slide. But what I want you to pay attention to are all these tiny blue specks scattered throughout the sample. Those are the kinds of things I'm comparing from one whiskey to the next. In one sample, carrying more microscopic material than another, I don't know, what do you think? As we zoom in, you can really start seeing how evenly these tiny blue particles are spread across the sample. Now remember, you can't actually see the alcohol under a microscope. What we're seeing is microscopic material that's suspended in the whiskey, and the methylene blue helps really bring it out into view. So now take a look at this. You notice how some of the blue material is starting to collect in certain areas instead of being spread out evenly? That's completely normal. Tiny particles and compounds can move around as the liquid settles on the slide. That's one of the reasons every sample develops its own unique pattern under the microscope. So now we're at 1,000 times, and you can really see how the sample is made up of tiny little pockets surrounded by microscopic material. I can't tell you exactly what every particle is just by looking at it, but that's not really what we're trying to do. I'm looking for patterns. Does one whiskey leave behind more microscopic material than another? Does it spread out differently? So that's what makes these comparisons so interesting. Okay guys, last sample before we get to the nematodes, but here's sample D at 40 times, and right away this sample looks a lot cleaner than some of the others we've looked at. I'm not seeing nearly as much as the blue stain material scattered across the slide. That doesn't mean it's better whiskey, but it does make it interesting to compare against the other samples. So as we zoom into 100, this little dark strand caught my attention. It looks more like a tiny fiber that ended up on the slide during the preparation than something that actually came from the whiskey. That's one thing you learn pretty quickly with microscopy. Not everything you see belongs to the sample, so it's important not to jump to conclusions. So we're up 400 times now, and this one's pretty clean slate. Even at higher magnification, the sample looks, clean. There are a few tiny stain particles here and there, but compared to some of the other whiskeys, I'm not just seeing much suspended material. And that's exactly why I like comparing samples side by side. So finally, let's get to the nematodes and see how they fare with these 4 whiskeys. You guys ready for this? Okay guys, before we get started, let me introduce you to the star of today's experiment. This tiny worm is called Caenorhabditis elegans, or C. elegans for short. It's a free-living nematode that's only about 1 millimeter long, but don't let its size fool you. It's one of the most studied animals in science. These little guys spend their lives feeding on bacteria in the soil and in my lab. They're eating E. coli, which is their standard food source. Scientists have been studying C. elegans for more than 60 years because they're transparent, they're easy to grow, and they respond quickly to changes in their environment. Nematodes as a group have been around for hundreds of millions of years, and today they're found almost everywhere on Earth, in soil, fresh water, and even the ocean. Now let's see how these tiny worms react when each whiskey is introduced. So first, take a look at this. Those oval structures inside the middle of the worm appear to be developing eggs. Most C. elegans aren't female. They're actually hermaphrodites, which means a single worm has both male and female reproductive organs. Once they reach adulthood, they can fertilize their own eggs and lay hundreds of offspring during their lifetime. What's really amazing is they go from a single egg to a fully grown adult in only about 3 days at room temperature. And that's one of the reasons scientists use them so often in research. And even though this worm is only about 1 millimeter long, scientists know the fate of every one of its 959 body cells, making it one of the best understood animals on Earth. Okay, here comes 1 drop of sample A liquor, hard liquor, right on top of them. So here it comes, guys, watch. And boom, right there. So let me focus, let me find out where they're at, let me move over a little bit. And they've just now been subjected to sample A's whiskey. Let's see how they do. So I zoomed in a little bit to get a closer look to see what's going on inside. So you can see the adults, the eggs inside of them right there, which they reproduce very fast, like I told you. And so it looks like they're slowing up. Did you guys notice that? Of course, right? They got 10,000 gallons of sample A whiskey poured on them. So I think if I would have had that done to me, I'd be out. So, okay, so I zoomed back out and yeah, for sure, they're slowing down. There are many that are still active, as you can see. A lot of babies in there. Yeah, I feel kind of bad, but you have you have an adult right there still wiggling around. Must be Captain America. But anyways, yeah, significant drop in movement. Here's the thing, guys. I watched them for about a good half hour, right? And they slowed down a lot. And then, believe it or not, they either woke up or came to because they started become active again. It's amazing. So I dropped all that on them and they came back to life. Okay, so I think we're ready for sample B nematode test. So here we go, guys. Okay, here we are, sample B, and not as many on the slide here, but we're good to go. Here comes the solution on the left side. So watch the left part of your screen and watch the reaction and compare them to sample A and sample B and see if you guys notice any difference. Good, there it is. So just watch them and let's have a look. And right away, guys, I can tell they're slowing up big time, faster than sample A's. So let me go ahead and speed it up and see where these are. So I zoomed in a little bit and yeah, they slowed up big time. You can tell they're not moving around quite as much. And as I watch them, it only takes a few minutes. So, okay, I think we're ready for sample C. You guys ready? Let's do this. All right, here's sample C nematode and busy slide here, you guys. So sample A, okay, sample B. So look at the difference. And what's crazy was sample A kind of woke up again after a while and sample B didn't. So here's sample C. Let's take a look and watch the part of your screen. I'm gonna introduce sample C's whiskey. And there it is. Let's see how they do. There's sample C's whiskey, just introduced. Same results, I think, as sample B. Yeah, of course, they're traumatized. It's a lot of liquor at one time. So yeah, they are slowing up a little bit, I can see. And let's zoom in a little bit, take a closer look. There we are, a little better, but yeah, they're not moving as much. We've got a little more action over here to the left, but they're having a hard time. Yeah, right here is where I introduced the beginning start of the whiskey and the solution that they were in. So there's the line right there. And let me zoom in a little bit so you get a clearer picture. But yeah, what do you guys think? Looks like they're not moving as much. There are a few, like I said, that are still wiggling around, but overall, yeah, they're gone. Okay, I think we're ready for sample D nematode test, and cast your votes in the comments now, guys. Guess which liquor or whiskey A, B, and C is. And here we are going to sample D. Here we are, a very busy slide again for sample D. Let me zoom in a little bit and get a closer look. Yeah, it's pretty amazing. Look at this, like a bunch of spaghetti, man, just everywhere, noodles, insane. So here we go, I'm gonna introduce sample D's whiskey. Let's take a look and see how they react. Look on your left side of the screen. There it is right there, guys. Wow, see that? That's a lot. It's like 1 million gallons of whiskey for them, but yeah, they got hit hard. So guess which one this is, sample D. And looks like they're doing okay so far, not too bad, but let's zoom in a little bit. That's a little better. So see the adults right there? A lot of adults. There's some babies in there too as well. So it looks like they're slowing up. It only takes time before this hits them hard. So let me zoom out and get a clear picture of what's going on here at the scene. Yeah, look at it. They're slowing down, of course, and there's still some one of the super strong ones there. They're still going, but I watched slide for a good 20 minutes, and eventually they all come to a halt. So anyways, so what do you guys think? Let's leave the microscopic world, head back to reality, and I will reveal sample A, B, C, and D. Cast your votes in the comments. Here we go. Well, guys, after looking at all 4 whiskeys under the microscope, it was finally time for the real test. Sample A turned out to be Jack Daniels. Yeah, a lot of bad memories with Jack Daniels. The nematodes slowed down at first, but after a while, they woke up. I couldn't believe it. Never seen that. I wasn't expecting that. All right, guys, sample B was Bulleit. Another bad experience with that. But over time, every nematode stopped moving eventually, and I never saw them recover. Poor guys. Sample C was Maker's Mark. What do you think about that? It produced almost the exact same result. The nematodes eventually stopped moving, I know that, and they never recovered. And finally, everybody's favorite was the dreaded Fireball. And another bad experience with that. The nematodes stopped moving completely. And after I never saw them recover, now remember, this doesn't tell us which whiskey is good or bad, all right? And it definitely doesn't tell us what would happen inside the human body. It simply shows how one tiny organism responded under these conditions. And I thought that was pretty fascinating. Oh, before you go, I get a lot of comments asking what microscope I use, what equipment I recommend, and how I actually do these experiments. But that's exactly why I put together my official lab field guide. I wrote it for beginners, so you don't need no PhD or a science degree to understand it. Just have fun. It covers the equipment I use, the slide preparation, the staining techniques, the microscope settings, and a lot of the tips and tricks I've learned along the way. And the best part is, every time I add new experiments or new techniques, you'll get all of the updates completely free for life. If you'd like to check it out, there's a link waiting for you in the description below. You guys, thanks for watching. Be good to yourself and others.